tuning in to our presentation about amyloid fibrils and how these specific unique proteins work, specifically in the human body. This presentation was created by Donovan Peterson, Alan Ramos, Erica Vargas, and I'm Elena Russert. Amyloids are abnormal proteins, and their polypeptide formation relies on misfolded proteins to form aggregates or clumps. These misfolded proteins are made from amyloid precursor proteins, shortened to the acronym APP, which is a type of transmembrane protein, or as we learned about it in class, integral membrane proteins. And these are produced largely in the brain. These amyloid precursor proteins are misfolded due to cleavage by different enzymes, and a suspected reason many neurodegenerative diseases are associated with amyloids is due to the metabolism of the amyloid precursor protein changing over one's lifetime. And I really like the graphic on the right because it shows just how beta amyloid proteins accumulate when the amyloid precursor proteins are cleaved incorrectly. The formation of amyloid proteins is caused by the presence of different enzymes that cleave in the incorrect spot. In this image on the right in graphic A, alpha secretase and gamma secretase work together to cleave at the right spots on the amyloid precursor protein, which produces a chain of hydrophobic peptides called P3 fragments. In the amyloidogenic pathway, however, beta secretase cleaves at the N-terminus on the amyloid precursor protein, which then causes gamma secretase to cleave at the C-terminus. And this chain of events leads to the formation of beta amyloid proteins. After selective enzymes cleave the APP to form amyloids, they can clump together to form long fibrous chains and transform a previously soluble APP to an insoluble plaque of beta amyloids, which are composed of several organized beta sheets, and in between those beta sheets are rows of hydrogen bonding. And both of those features contribute to the stability of the protein and consequently make them extremely hard to break down in the body. Additionally, there is no cure for amyloidosis or the associated diseases at this time, but it's been shown that a good diet and exercise can decrease symptoms of some neurodegenerative diseases such as Alzheimer's or Parkinson's. And in general, diseases are treated case by case, and there are a ton of pharmaceutical companies working to create therapeutic drugs to treat these amyloid-related illnesses. In this study, several germline sequences from humans were analyzed, and researchers discovered that different somatic mutations had an impact on the stability of the amyloid proteins present. For example, when various monoclonal immunoglobulin light chain proteins reached their unique melting point, 50% of the protein unfolded. And when the protein unfolds, it can no longer contribute to the production of amyloids. So some proteins like the AL or light chain T05, for example, required a higher melting point of 50.3 degrees Celsius to unfold 50%. But in ALT03, it only required a temperature of 29.8 degrees Celsius, which is about 85 degrees Fahrenheit, which is way under the temperature of the normal human body. So it's most likely that this protein would never form amyloids anyways.
Hi everyone, my name is Donovan Peterson, and I'm going to begin on slide 6 talking about the location in the human body that is affected by amyloid proteins. The organs that are affected, beginning at the top, are the heart, kidneys, liver, spleen, nervous system, and digestive tract. I have an image of heart cells in purple shown on this slide, and the bluish light gray color is shown as the amyloid deposits between the heart cells. Now we can see if this is left untreated, the severity that it can cause to the actual organ itself. If the plaque continues to accumulate, the heart's not going to be able to function as it should, which has life-threatening results. Mayo Clinic, in an article that they wrote, stated that patients of this degree, if left untreated or undiagnosed for a period of time and let this accumulate, most of the patients would benefit from either an organ or a stem cell transplant. On slide 7, we can see a picture of a human body. On the left-hand side, we have images circled in green. On the right-hand side, we have images circled in red. Beginning on the left-hand side, we can see that the functional amyloids, these are amyloid proteins that are helpful or beneficial to the human body, and they help with the memory formation, hormonal release, skin pigmentation. And on the right-hand side, we have diseases caused by the amyloid plaques. Beginning at the top, we have neurodegenerative diseases, cataracts, non-neuropathic system amyloidosis, type 2 diabetes, and bone marrow. On slide 8, I'm going to finish up talking about the demographics that are affected by amyloidosis. Studies have been done that are shown that most of the people that are affected, or the ages that are affected the most by this disease, are falling between the ages of 50 and 65. There has been rare cases that a patient was 20 years upon diagnosis, but that's pretty rare. This disease is also more commonly found in men than in women. The onset of the condition is sometimes caused by an underlying disease that produces an abnormal protein, which is inherently amyloidogenic. It can also be formed by a great increase in normal protein being produced, which has the potential to form amyloid proteins.
Amyloids are formed through the polymerization of hundreds to thousands of monomeric peptides, or proteins, to form long fibers. These are formed by soluble proteins, which assemble to form insoluble fibers, which are very resistant to degradation. One method by doing this is by fragmentation, which is when the fibril breaks down to 2 or more shorter fibrils, which increases the number of fibril ends that allow the recruitment of new monomers or oligomers, therefore accelerating the fibril formation. So these can be caused by misfolding or by mutations in these regions, causing them to self-assemble into aggregates or big groups. These structural rearrangements lead to formation of amyloid fibrils. There are 3 phases. During the lag phase, it's mainly protein monomers and oligomers, which is followed by a rapid growth phase in which protein fibrils are formed, followed lastly by a plateau phase in which the reaction is ended due to the depletion of soluble proteins. So here we have a structure of a 2D amyloid. White is the main carbon, yellow is the side chain carbon, blue is nitrogen, and red is oxygen. Amyloids contain beta sheet stacking interfaces, which are sometimes called dry or wet interfaces. The dry interface lacks water molecules and has a steric zipper, which are pairs of self-complementary beta sheets that are formed and are considered the backbone of amyloids. The other interface, the wet interface, is composed of hydrogen bonds between side chains, and some are via water molecules. So the exclusion of water molecules between some of the beta sheets maximizes the entropy gain for the formation of amyloid fibers. Followed, we have the 3D structure of an amyloid. In between the beta sheets, there are many rows of hydrogen bonding. This hydrogen bonding can increase the stability of the molecule and makes amyloids very, very stable molecules. The cross beta sheet pattern is considered a diagnostic characteristic of amyloid structures. Each filament consists of cross beta structures and may be formed by beta sheets stacked on top of one another. These strands can be arranged in anti-parallel formations, but are most often found in parallel beta sheets.
Hi everyone, my name is Erica Vargas, and today I'll start by talking about the structure of amyloids and their function. The cross beta sheet structure of amyloid fibrils provides a stable structure for the formation of hydrogen bonds between the fibrils, and the fibril itself is made up of 2 to 6 protofilaments, and you can see this in the image to the right. The more protofilaments there are, the more tightly the filaments can wrap around each other. Amyloids can be produced through different proteins found in the human body, and it is common for proteins with many large aromatic side groups to form amyloid fibrils because of the pi-pi stacking that occurs. With this being said, hydrophobic side chain interactions can also help fibrils self-assemble. Amyloids
possess a left-handed fibril twist due to the many beta sheets that are found within the structure, and this makes it difficult for proteolysis to occur. This overall structure allows for plaque buildup within the human body because of how stable it is. Unlike regular misfolded proteins, amyloids are different from them due to their distinct cross-beta alignment made from fibril-forming monomers. These fibril-forming monomers self-assemble to make up the quaternary structure of the amyloid. The beta strand structure uses side chains to form two parallel beta sheet structures and a hydrophobic core. These two components help to stabilize the overall structure. And due to the cross-beta alignment, fibrils tend to be double-layered, and this is demonstrated in the figure below. There's beta sheets at the top and beta sheets at the bottom, and they are going in opposite directions. After doing this project, we found that there were a lot of diseases that were associated with amyloid production in the human body. There are three major forms of amyloidosis. Primary amyloidosis is the most common and is not usually associated with another disease. It affects parts such as the heart, lungs, intestines, nerves, skin, and tongue. Secondary or acquired amyloidosis is associated with chronic diseases such as tuberculosis, osteomyelitis, or rheumatoid arthritis, and it commonly affects the kidneys, liver, spleen, and intestines. With secondary or acquired amyloidosis, once the chronic disease is treated, the amyloids tend to go away. With hereditary amyloidosis, it affects the nervous system and the digestive system. The figure on the right shows a normal brain compared to the brain with Alzheimer's. Alzheimer's is one of the diseases that is caused by amyloid plaque buildup, and you can see in the figure that the neurons may have trouble signaling each other due to the plaques that are located between the neurons. Symptoms range from person to person depending on what organs are affected and can cause many complications. The way to test if a person has amyloids can be through the blood and urine, and this can see if they have abnormal protein production in the body. But the most reliable way to take a sample is through a person's skin, liver, kidney, or gums, and they will stain the sample with a dye that reacts to amyloids.